Hi, my name is Dr. Stokes Peebles, and I'm in the Division of Allergy and Pulmonary Critical Care Medicine here at Vanderbilt. Our lab studies lung inflammation, and we're particularly interested in two different environmental factors that cause inflammation. One is allergy, and the second is viral infections. In regard to allergy, we have several different projects in which we're trying to downregulate allergic airway inflammation as a potential treatment for asthma. As you know, asthma affects roughly 5 to 8% of the United States population, roughly about 20 million people. And the therapies that we have for mild asthma are very good, but for those of severe asthma, not so effective. So what we want to do is we want to come up with novel medications that we can use to treat allergic airway inflammation and that may have an impact on asthma. One of the things that we're very interested in is a lipid product called prostacyclin. Prostacyclin is made by the endothelial cells, or the cells that line the blood vessels. And for a long time, it was thought to only have effect on the vascular system. However, in our lab, over the last 10 years or so, we've understood that it has very important immunoinhibitory properties as well. It downregulates dendritic cell function, the ability of dendritic cells to present antigen to T cells to drive allergic responses. It also has a negative impact on T cell cytokine production. Cytokines such as IL-5 and IL-13, which are very critical to the allergic phenotype. We've also found that prostacyclin negatively regulates the group 2 innate lymphoid cells, or the ILC2 cells, which have only been discovered over the last 5 years. So prostacyclin has been known in treatment for about 30 years now, predominantly for pulmonary hypertension. However, we feel that this medication could be repurposed for the treatment of severe allergic diseases such as asthma. One of the other projects we have that relate to allergic inflammation is looking at glucagon-like peptide 1 receptor agonists. Now, glucagon-like peptide 1 is something that we all produce when we eat. And what happens is that glucagon-like peptide 1 is produced by the endocrine cells of the large and the small intestine, and it acts on the pancreas to elicit release of insulin. And this helps control our glucose level. Well, what we found is that it also has an impact on allergic airway inflammation, really a surprising finding. So Dr. Kevin Neiswinder, one of our collaborators in endocrinology, had been doing some research on GLP-1, or glucagon-like peptide 1, and he found that in several animal models of autoimmunity, it downregulated inflammation and actually protected against autoimmunity. So he came to us and said, why don't we look at this in regard to allergic airway inflammation? So we've done quite a few experiments. Shinji Toki in our lab has been the primary one doing this, and he's found that GLP-1 receptor agonists, which are FDA approved for the treatment of both obesity and type 2 diabetes, negatively regulate the ILC2 cells and also downregulate T cell cytokine secretion that's important for allergic airway inflammation. So we have a grant currently under review at the NIH, which we hope will be funded so that we can perhaps use this in the treatment of people who have obesity and asthma. The last area in our lab that we focus on is viral infection, and particularly respiratory syncytial virus infection. Respiratory syncytial virus, or RSV, is the main cause of infant bronchiolitis in the United States, and roughly about 100 to 150,000 children each year will be hospitalized with RSV bronchiolitis. It's also a very important cause of asthma exacerbations in the winter months. So what we've been doing is trying to figure out what part of the RSV genome is responsible for this asthma-like phenotype that causes the symptoms of shortness of breath and wheezing that are classic for asthma. And what we've determined is that there are certain sequences in the RSV-G gene which seem to modulate the host immune response that makes it more mucogenic and also causes airway responsiveness. So what we're doing now is looking at ways of modifying this. And we've just recently received a 5-year grant from the NIH, the Asthma and Allergic Degrees Cooperative Research Center, to help study these things. And so we're very interested in this. So these are the major focuses of our lab, both allergic airway inflammation and also virally induced airway inflammation. And we study these things as they relate to asthma. So if you're interested, I'm happy to talk to you more about it at any time. Thank you very much.